Hi everyone, this is Aurelie Najm from Glasgow. I am here in Copenhagen for day 2. Again, very exciting science. You might hear some background noise because I'm still in the conference center, but I had to share all of these exciting things with you. So, 2 abstracts I want to really talk you through today, and both is OP0084 and 0085. You will see that these titles can sound very complex at first, especially if you don't do any translational research. But this is what I thought it would be really good if I could take a few minutes and try and demystify these, because they are exciting, but also they are very relevant for what we do in clinical practice. So OP0084, digital spatial profiling and how it reveals distinct macrophage transcriptomic signatures in array. So I'm going to give you a little bit of background here. So this cohort is a cohort of patients in remission. And the issue we have usually in clinic is that patients who are in remission, some of them relapse and some of them don't, and we don't really know why. And so what this group has been able to find out is that in those who relapse, actually, they have subpopulation of macrophages that are present in their synovial tissue that are actually absent in those who not relapse. And based on these elements, the idea is to better stratify patients. However, if you look into synovial tissue of 2 patients, one that will relapse and one that will not, you won't be able to see the difference if you look through a microscope, because the histology would look the same. So what is digital spatial transcriptomic? Actually, this is a technique that allows to look into the tissue histologically, but also to select different areas of the tissue where you're going to do specific transcriptomic analysis. And in doing that, they actually realized that in areas that are exactly looking the same, they were able to identify different macrophages subpopulations. And these populations that are responsible, that were associated at least with relapse, they found them in relapse through this technique. So this is quite exciting, and this is promising for the future, because we can imagine we can also look at so many different types of cells, lymphocytes, etc. So that's the first abstract. The second one, OP0085, is kind of reinventing analysis of the R4RA trial. I don't know if you remember this trial. It was quite exciting. It was last year as it got published in the Lancet. So what this group from London did as a very big collaborative effort is they did a randomized control trial where they stratified patients that were resistant to TNF inhibitors into rituximab or tocilizumab based on how their synovial tissue looked. And while histology was it was a bit of a deception to see that histology really did not help predicting what patient was going to respond to what treatment, they were able to identify molecular signatures associated to tissue rich in B cells or tissue poor in B cells, and patients that had a tissue poor in B cells were more likely to respond to tocilizumab, while the other ones weren't. The issue though with this is that, see, at approximately 30% of the patients that were B cell poor phenotyped also did not respond to tocilizumab. So the prediction was about 60%, which is much better than what we do. But obviously the question was, can we do better? So they went back to these patients, they went back to the tissue, and they did some more deconvoluted kind of analysis where they looked at cell lineage specific transcripts, and they were able to better subcategorize these patients. So all in all, very promising, very exciting, and it sounds like it's very fancy analysis, and actually it is, but this is really what is going to take us to the next step, and I'm sure it's going to really help us a lot to stratify patients in the future, and it answers very common clinical questions that we all have. So stay tuned, connect on remnow.com for more content, follow me on Twitter at Aurelie Najm, and see you soon.